I am Joanne Geer. I've gotten a chance to say hello to many of you this morning. I'm the Executive Director of the Biopharma Research Council. We are an association for scientists across all the different silos of biomedical research. So that's industry, academia, nonprofit, government, and suppliers, and those who love them, which is who's in the room today. We look at themes that allow us to address different ways of bringing people together, because our mission is to get scientists together in ways that they might not otherwise find each other. So just in this past year, we had a CRISPR conference in Boston. We had a microbiome program at the National Cancer Institute in Maryland, and a Internet of Medical Things focusing on cybersecurity and discovery and development in New Jersey, where our office is. I'm going to introduce one of our committee members who's been a big supporter since the beginning. We're not going to do long introductions to everyone because almost everybody's bios is in your program, but Winona's is not, so I'm going to introduce her now. Winona Satcher is an urban designer, landscape architect designer, and city planner. She's a graduate of Auburn University College of Architecture, Design, and Construction, and moved to North Carolina to work in professional design firms in the Research Triangle and Durham. In 2008, she started her first urban design, economic development, and leadership startup. Through her second startup, which is iDrive Change, which she's doing now, she combines landscape architecture, systems design, community development, and innovative economic models to facilitate equitable revitalization programs through various collaborations and partnerships. She has written for the Huffington Post, participated in the Aspen Institute, Bloomberg Philanthropies National Innovation Program, and was a Vanguard Top 40 Under 40 Vanguard through Next City. Winona.
Thanks, Joanne. Good morning, everyone. So why am I here? There's nothing in my bio about biotechnology. I am here, like Joanne says, a long supporter. Wanted to welcome you to the Triangle Biotech Research Symposium today. Joanne mentioned in my bio that I look at systems design and really interested in community engagement and how we bring different systems together. So this makes it a perfect space for me. I will say that to our speakers, welcome, and to our audience, welcome. Today we have a program filled with, as you can see, amazing speakers to help us understand and build on the idea of convergence. Love that word. And the idea is that looking through various aspects of what you do, community engagement, bio and technology, research and development, as well as business development. So we're stretching the conversation just a little bit today. Often our dialogue is centered around one of those specific groups, either the research component or the development component or the education component. We are adding entrepreneurialism as well to the conversation. We'll share with you as you hear our speakers and network in the lobby between now and the end of the conference, that we will touch on the application of the technologies that we talk about in our classrooms, that we research and develop in our labs, and then how do we go from there to the other spectrum of actually entrepreneurialism and startups and the application of what we have learned and researched and the methodologies and strategies that we implement. So without further ado, without taking up too much of your time, I just wanted to say welcome. Glad that you all are here. Wonderful day outside. No more rain. Yay. No more fog. Yay. Welcome to Durham, North Carolina if you are from out of state. And we will get started.
I'm Dick Kouri. I'm the chief evangelist for SIMS. And Paul and I worked together about 10 years now at NC State University. And I have to admit, what Paul's brought is a kind of formal structure to innovation management, and you're going to hear it in a minute. And that structure allowed me, I was an entrepreneur and practiced entrepreneurship for a long time. My problem was I didn't really have the words to describe what we were doing as well as Paul did. Paul came up out of IBM and actually was pretty much the chief innovation officer when IBM turned the ship around back in the late 90s. And that was a big ship, to say the least, to turn around. So much so that when IBM, I guess it was 3 years ago, they went through their 100-year anniversary, and when they did, they invited back the 100 most influential people in IBM's history, and Paul was one of them.
That we're still alive.
There were some, yes, verticality counted, that's true. And, but that is in fact part of there's not many people that could bring that kind of gravitas to the event called, or the structure called innovation management. Most of us think of it as a more ephemeral sort of term, but it can be managed, it can be measured, it can be taught, and can be made better, and I think we're going to hear it right now.
Oh. Where's my little flicker? Oh, here it is. You stole it, Dick. No, I got it. Good. Not yet. Well, this is like the size of an MBA class. I should ask you to all move down front like we do the students, right? No, we won't. But actually, the intimacy of the group is going to be better because you'll see there's some cases and examples that I'm going to give you that I'm sure you're going to be peering your eyes at and wanting to know more. So I'll leave time so that we can ask questions and have a dialogue at the end. So I won't be just blabbing at you for a solid, 45 minutes now. You can see that I've taken an African proverb to use as the centerpiece for my presentation. Is everybody familiar with "It Takes a Village"? We're not talking about the Democrat candidate for the presidency and her great book. We're talking about a really almost ageless African proverb. And what is that proverb? How does it end? How does it complete? It takes a village to do what? Raise a child. Okay, we're going to replace the child with an innovation, Dick. We're going to use this metaphor to talk about innovation and how to create better innovations. And this will weave its way through the whole talk. We went through this. Okay, CIMS, a little plug for CIMS. The Center for Innovation Management Studies is what CIMS stands for. And it's a 31-year institution. It came out of the National Science Foundation. And to our knowledge, it's the only research center devoted to how to innovate. Now, I know you're all interested in what to innovate, and I'm a techie too, just a different type of techie. I'm an information techie, where you all are biotechies. We love to talk about what to innovate, but we're not going to do that. We're going to talk about how to innovate, the process of innovation, a little bit with you this morning. So it's a great center. It's now graduated. We're not on the NSF dole. We're not even on the NC State or the state of North Carolina dole. We're completely self-sufficient. We're attached to the business school down at NC State. We're chartered at the UNCGA level. So that's a little bit about CIMS. Maybe if you have questions, we have, where's Michelle? Wave your arm. She's the real boss, the managing director of the center. And we'll all be available at the first break if you want to know more about CIMS, okay? Oh, we got new offices too. We've expanded to Preston. I'm sure will fill your ears up at the break about the frontier, right, Preston? This is really a cool thing. It's going up down the highway from you off of 54 in Davis. It's eventually going to be a 100-acre, $2 billion project that purports to be the biggest or largest open innovation center in the world when it's completed. So I thought it was very appropriate, given my talk, that we give a plug to Bob Jolis there in the center and the Frontier, where we're located. Here's my agenda for the next, let's see, let's make it about 25, 30 minutes if we can to give you time for questions. We're going to briefly talk about what is innovation, what is open innovation, how do they differ. So I'll give you some basics of both, some frameworks that you can take away with you, and then I'm going to talk more importantly at why it takes either open or just standard innovation, how it takes a village to actually succeed at innovation. And then the main topic today is how we're using big data analytics. You can see from my collage that I made up, we're going to be talking about all these topics, how we're using big data to actually connect the village, converge scientists and people involved in commercialization, and use analytics. I think you'll find this very interesting, how we use it to actually connect that village. And then I have some final thoughts, and most importantly, your questions. All right, let's talk about what innovation is not. You can look at this wild scientist. Perhaps some of you have him in your lab back at the ranch, or people like him. This is not innovation. This is, I want to start with what it's not. Innovation is never, ever done by a lone genius. Invention may be. Innovation's much tougher, and it requires people of all skills, backgrounds, nationalities, and genders to do it really, really good. And that's going to be right through my theme as we talk this morning. Here's a nice one. Here's another metaphor. I love metaphors and stories. Hargadon, who is a Sims researcher out at UC Davis, it has a wonderful way with words. I love his words. Innovation is like trading knowledge. It's what went on back in the medieval bazaars, perhaps Marco Polo era, 1290 or so, where East met West, and ideas actually built on top of ideas to create really great innovations. Does that sort of resonate with everybody, that it's a social, huh? It is. At the heart of all this, we got all kinds of tools and formulas and techniques and big data, but at the heart of everything, it's a social process. Pop quiz. I said you look like an MBA class. Is this innovation? I'm going to call on somebody. Look at this chart. I'll give you some facts. Now, Preston, don't you do this. Preston knows the answers, because I'm always throwing this pop quiz in with every talk. Huh?
And he will.
And yes, I will call on you, yeah.
And he'll call on you by name.
I don't care if we get through all the damn slides or not. I'd much rather have you jump up in your seats and be excited about something. Let me give you some facts while you look at this chart. In the first quarter of this year, there were more mergers and acquisitions in the pharmaceutical sector than in the whole prior year of 2014. So in the first quarter of 15, anybody have any idea how much we're talking about in mergers and acquisitions? Dick's trying to read all this. Dick, you could get it off the chart. It's about 260 billion. And that was greater than the entire year of 2014 that was accomplished. And there was something like 60 billion in the pipeline yet to be announced. So M&A is what many people in pharmaceuticals would equate to innovation. Right? Wrong. So we don't count M&A as innovation at all. And you can see my big red nose, and you can read the kick-out box. Most of these gambits are strictly about buying products and revenue streams. Then they haul out the organization that created them, and they lose the very thing that was important: the people, the knowledge, the great ideas. Another pop quiz. EMC and Dell just merged. Two big powerhouses in the IT industry. Who's going to be the winner? Oh, who said that? Yeah, why?
You have two monolithic Goliaths turning into one super monolithic Goliath.
Right.
That's not the way you do interesting, exciting new things, especially in IT.
Oh my gosh. Correct. Go to the head of the class. So we're doing pretty good. We got a couple A's already on the pop quizzes, right? My prediction is, and I know them both, Tucci and Dell. Tucci's the head of EMC. Dell, of course, Dell will be gone in 6 months. And then they'll start the inexorable trend of firing and merging people and restructuring the two corporations. Doesn't count as innovation. What is innovation? This is one of Sims's definitions. This one pretty much, where's Deborah Dougherty? She's written books on this, for God's sakes. We don't have to guess at this. Is that a good one? I think that's a good one. It's the process of converting ideas into legitimate, we'll get underscore legitimate business opportunities. Again, I'm from the business school. We're going to talk about the business of R&D this morning, not so much the science, but we will get into that. So what is open innovation, and why does it take a village? Okay, so you could add to this word chart. Open innovation shows itself in many modes, forms, models. These are just a few. You could probably add your own to this. This is when I'm talking about open innovation. I'm talking about structures like co-marketing, collaborative R&D, joint development centers, et cetera. Not M&A, but in general, a partnering model, all right? Keep this chart in your head to when I come back to how we train Watson to solve this problem. All these names and pseudonyms for what constitutes open innovation. Why would you pursue an open innovation business model in the first place? Here's, maybe you know this fact. Most all big ideas, radical, disruptive, come from outside the boundaries of the company. Look at this chart. We had a special SIMS conference. We just completed one earlier this week for 2 days, talking about open innovation revisited. This is one we conducted at Purdue University. What was it, 2006, I think is on the chart. And we had a great professor, Dr. Sue Reed, and her buddy Dee Brittani, and they did a lot of studies on innovation. And it's a fact that the big ideas that lead to disruptive innovations almost invariably come from outside the company. Does that sound like a riddle? What? Does that make sense? See that big information flow arrow? For incremental innovations, it's just the opposite. You know, small feature changes, product design changes, whatever. Information flows from the center out. But look who makes up the macro environment. This is the beginning of that village that I keep referring to. Connecting them to the company is everything if you want to do big innovations. Okay, I guess, Deborah, we can give old Hank, I don't call him Hank to his face, Henry. He's a good guy. I have talked with Hank. Hank's out in, what? Henry Chesborough. Yeah, he wrote some really foundational books. Come on in, sir. You were stuck in the traffic. Come on down. Yeah, because I was too. I left home at 6:30. Come on in. Yeah. Yeah, come on down because I haven't called on you yet. So there, there is a little pop quiz. Henry is a genius, and he really studied. Now, this is a very complex picture. Look at this funnel with holes in it. Everybody's seen the metaphor of the funnel for representing the company's R&D process, perhaps. Look at all these holes stuck in it and things coming from outside in and from inside out. What he was trying to do in this drawing, and he actually did create this drawing, is get the notion across that all the ideas don't have to come from within. Again, if you want to do big stuff, bring them in from the outside. You may use it again in your own core businesses and markets, but then you may elect to start entirely new markets or spin it out so it can flourish on its own and under entirely different structures. That's what that picture is trying to depict. It's a more complicated, for sure. Come on down, sir. The pop quiz. Have you read your homework? No, I'll embarrass everybody that gets off of. No. He drew this picture, but this is the key idea here. In this information-rich environment, companies can no longer afford to rely entirely on their own ideas. This information-rich environment. We're going to come back to that, as of course you know, if you've been looking ahead. Okay, this is some interesting work done by a group called the Industrial Research Institute of the United States. They're the most research-intensive firms of our land. SIMS is a member or group affiliated with them. And they did some nice research on the evolution of what's called co-creation networks or open innovation. And actually, they precede Henry. I hope Henry doesn't take any offense to this, but open innovation has really been going on quite a long time. When companies found, perhaps GSK across the street found out that the same processes and skills for managing research across their campuses could actually be applied, as you're seeing, outside the company. Because as companies got larger, the R&D center, IBM used to have one. I think they've got 6 or 7 or 8 major ones now, all over the world. So the same skills that it takes to coordinate those campuses could actually be leveraged and used to work with other entities. And that goes back to probably the 70s or 80s. Dougherty can keep me correct on all these dates, but probably that was the first inklings of open innovation. Then it got really going when they started including in the village these ideas of universities. Wow. That was a big step. I'll assure you, I don't know if GSK had met anything like NC State in its life. Do you think so, Fran? When it started connecting with those members of the village and government labs, of course, and then later expanded to alliances and joint ventures and whatnot. Most recently, and perhaps most importantly, they include customers in the village themselves. Because if you're trying to develop something that is a radical or breakthrough, and you try and do it without customers or any kind of connection with the markets, you'll fail. I can assure you. And if you're in the services and solutions business, you shouldn't even start until you've engaged with lead customers. Now, here's the big caution sign. Dick, is that crack in the ice and danger sign big enough? No, it's not. Because here's the big problem. The responsibility for forging these villages typically falls on the R&D function. Question group, are they, in your mind, some of you are members of that, are they equipped to do that? No, speak up. Yes, some are.
Well, it seems to me as much as an issue of capabilities, it's an issue of knowing what they have permission to do.
Yeah, that's a good, go ahead, say something.
Implicit in that one
Yeah?
is allowing to do.
Right.
Now, that speaks to the culture of the organization, and that's probably the biggest reason why it has to be a function of the corporation,
Right.
not a function of a particular area. R&D or customer service or marketing or management, all of them have equal.
Right.
So the excuse comes that, the funding, the money part also.
Oh, the money part. Talk to me about the money part.
Exactly, because then you have priorities,
Right.
This is our priority to push this compound to phase 3.
Right.
Everything else stops.
And-
Keep your ideas, keep running whatever you are doing in the lab. Don't
Right, right. You've been there, I can tell. Right.
Until the phase 3 is successful, then we get ready. So.
And if we have a horse race between two competing ideas, and one's from inside the firm and one's from outside, in a political world, which one's going to win, right? The inside. Right, so it takes a great deal of leadership, thought, strategy. This is closely tied to strategy. It also takes new skills. We actually have research streams, and have you ever heard of boundary spanners? We've actually done in SIMS big research into the kind of people that have these special talents for spying ideas and then connecting. We do. They're called boundary spanners, and how to cultivate them in your organization. Pretty soon I'll show you a boundary spanner on steroids, but anyway, this is the problem. Now this, honest to God, Carmen was looking at my slides. Carmen is my wonderful wife, and she said, Who said that? What did they, who said, Dick was there. I cannot mention the corporation, Fortune 20, probably 10, head of R&D. Why would I want my researchers to understand the market? How long did it take before we jumped him?
A while. I couldn't believe he said
Yeah, Dick was aghast. No, this is a direct quote. Why would I want my researchers to understand the market?
Is this company successful?
No. They're extremely rich. $1 billion run rate a year, no products in 11 years. I wonder why. Huh? Don't try and guess. I'm not going to tell you. Well, maybe I could be bought off. No. But this is not a one-off example. We hear this all the time. Why would I want my researchers to understand the market or some form of that? All right, so let's go into how big data can help connect the village, all right? Do boundary spanning on steroids. All right, so every presentation, if you know me, always contains this chart. Always. I don't care what the topic I'm speaking on, I always stick the value, don't I? Michelle knows, it's got to go in. The valley of death. Who knows about the valley of death? I got you drawn on here. You all live in it. See, that's you. Now, what's so ominous about being you in the middle of that? What does that represent? Huh? Yeah. Yeah, what we've found through research, not just SIMS, but a mountain of research is crossing that valley, connecting people on both sides of that valley, i.e. creating a village, is the hardest damn thing in the world. It is, and it's where most good ideas, you're shaking your head, they crumble. There's all kind of bones down at the bottom. The bones of good ideas, Deborah, that are scattered around the valley. Because bridging these two communities, I'll call them, is very hard. And it gets, this valley exists inside of firms. IBM has this valley. GSK has this valley. Now just take the discovery mountain and push it outside the company. And everything is factorially harder. Come on down. I know you were stuck in the traffic problem out on 147. Right. Anyway, more than just a resource problem that came up, it's also a very deep cultural and information divide. They have very different value systems, people involved in discovery. How many of you are involved in commercialization? Oh, more than I thought. How many in discovery? We have both sides of the valley equally represented. Good. You think they respect each other? No. No, they don't. No. What do the people in marketing and sales call the engineers and researchers?
Actually, I love them. I have nothing but respect, but I don't think they respect me.
So which side of you are you with?
I'm on the commercialization.
What do you think they call you? They have nicknames for you. No.
I've got no business knowing.
They do. They have completely different value systems. They're measured differently. They went to different schools. They even have a different language. They do. And so you try and put all this together into a village and have fun. All right, so this is probably why you came. We started using Watson before it was a product. IBM graciously gave us the Watson algorithms. Just for FYI, they go back 50 years. They're almost as old as I am, Kelly, for gosh sakes. The natural language processing algorithms that can actually read, and 20-some languages are decades old. that's sort of the heart of Watson, and IBM was gracious enough to give us the software before it was a product, well before it was a product. And then we did something different. We started connecting old Watson to the internet. And we broke every programming limit in it and caused brownouts in Raleigh when we attached it to supercomputers. So we have the same algorithms, but instead of spoon feeding it information about Shakespearean plays and rivers of the world so that it can compete with Jeopardy champions, we're answering these type of questions.
Who was Watson?
No, go ahead. No. No.
This was one of the first research directors of IBM Corporation, Watson Laboratories.
Right. It's the founder.
And the founder.
Right.
The founder of IBM.
Yes, sir.
It's not J.D. Watson.
Yes, in the back.
It's not the Watson and Crick?
No, it's not. It's not J.D. That's what I meant.
Yeah, so we've had these programs for about 8 years, and we're using them to answer these types of questions. Now, these are deceptively simply stated, as I'm going to show you in a moment. We're going to look at a couple of these as to how we train Watson to find viable partners to create a village. Okay, let's do a couple cases. First of all, we'll start with commercialization people. These are real cases. Now we've mussed them up a little bit, so you can't say, that's Merck, or that's, we know enough not to give you too much information, but they are actual cases. So please read the case, get this in your head. In the sake of time, you can read faster than I can talk to this. And you can see what they want to do. They want to rapidly add to their in-house skills and knowledge from outside their firm in a field they know nothing about. At least they had the presence of mind to recognize they didn't know anything about it. Some companies just keep going, but they see this huge trend coming of personalized medicine. Who hasn't? I'm probably going to be talking about it all day long. And they see it coming. They see the prospects of it being applied to horrible cancers, but they don't have the knowledge. They don't have the in-house skill and knowledge, so they want to partner. And specifically, the leading thought leaders, let that hang in the air, in the world. Now, this is dense. Don't worry, whoa. This is an actual script. This is actually what was fed to Watson. Now, it wasn't just, it looks logical this way. Well, it should look logical as I step you through it. But this is the result of many iterations. But you break down the main question for Watson into a set of sub-questions. And the first one is, which medical centers has NCI issued grants in excess of $1.5 million for developing personalized medicine for oncology? Watson can answer that question after you train it to answer that question. You train it as you're reading on by creating a set of objects or dictionaries so that it knows when it comes to medical centers. What do you mean by medical center, Mugge? Well, what do we mean is we just feed it a list from U.S. News and World Report of the top. We could have chosen any list, but just for the purpose of this exercise, we just took U.S. News and World Report. What are we also doing while we're doing that? We're curating the data, we're vetting the data. General thing we find, how many projects have we done? 100, 150? Web data is not real data. No, we hear it all the time. Because the paradigm is internal structured data, so this unstructured crap that sits out on the World Wide Web is not real data. I'll show you, it is real data. So we went to NCI, and we saw where they're giving major grants, and we just used arbitrarily. Was it a 1.5 actually that we set it at? Yeah. 1.5 million in this field, and then we developed some special macros, subroutines like money finder. So we're feeding all this to Watson at billions of operations a second, and the answer is that they've issued 52 spore grants to 52 medical centers. We have the whole list in 21 states. The states is important too because remember, given the proximity of the corporation in the case, they would much rather deal, if they were in Houston, with MD Anderson than fly to Duke. Both were issued spore grants, both great med centers, but the state and proximity is important. Next question. Which of those were focused on the particular cancers? And here we had a dictionary called oncology. Do you think we just trusted ourselves to define oncology? No. So where did we go? Who would we go to that has defined oncology? No? NCI, right? Or the National Institutes of Health's glossaries are magnificent. So rather than stumble, bumble around trying to define it, we just take the whole glossary of terms and plug it into Watson. What? Oh, I looked it up again. Special Programs of Research Excellence. Great question. I got up at 5 and did that.
Can I ask a secondary question?
Yes.
You used the top 50 U.S. News and World Report, which are doing oncology. Why not use the Comprehensive Cancer Centers from NCI as a list?
We could have.
Okay.
We could have.
And there probably is an overlap there.
Yes, we could have. But see, that's all part, the real jewel of this thing is getting people like yourselves around the table to have just that kind of questioning and discussion about the model before we feed it into Watson.
None of us trust U.S. News and World Report.
Oh, geez. Okay. And moving right along, let's look at some. Yeah. Anyway, we found 10. And then who were the key investigators? Now remember, the company's looking for skilled people. So that was actually easy to find once we had isolated the grants of a certain size at these medical centers. We could pick out the PIs, the co-PIs, the lab chiefs, everybody. 143 individuals. It's not 146 or 100, it's 143. Now, we went further. Now you're going to say, well, most of that could have been done if I'd had just used the search engine on NCI's site. Eventually, I could have hunted and pecked my way to death to find those 143. Yeah, you could have. But now see what we're using, the data we've almost automatically extracted. We go further. Which of them have been linked with startup activity? What are we why are we looking at that?
Experience in industry.
Speak louder, yeah, right.
Experience in industry, not just engaging startups, they have to learn.
Right. So we actually use Watson to create a data set. See the quotes around it? Now this is what you can't do with Google. So we actually created, we had the URLs of the top, I think it was 398 exactly. Dick always corrects me, but say roughly 400 life science focused VC firms in North America. This is a big data set. We just swallowed their websites, kaplunk. And now we go at that with the list of 143. And we can find out where they were, what they're doing, what's their start activity. Give them the punchline. How many of them were starting new companies?
Of the 143?
Yeah. None. None. There's reasons for that. If we have more time, we'll get into it. They were positioned on the Science Advisory Committee or board of startups and the VCs themselves. So the VCs recognized the knowledge these women and men possess, but they weren't actually launching companies. And then we went on to do more and more. There's no end to the, once you get this Lego kit built, you can go deeper and deeper and deeper. This is a general approach we call automated technology and market mapping using Watson. But I want to go the other way. This is probably even tougher. This is going from the discovery side across the valley to finding potential commercialization partners. You think schools are interested in this? Would you be interested in this, Fran Liegler? I know you would. This is right. Yeah, because these great universities around us have wonderful ideas and technologies. They just need to be probably connected to companies sooner that really see the full fruit of those efforts and inventions, right? So this is case number 2. We're going to go the other way across the valley. And how did we do that? Well, in this case, you saw what the small biotech had in their possession or thought they had. They were looking for drug companies that have failed phase 2 clinical trials in these cancers in the last 18 months. Everything that's underscored is important to the problem. Do you agree with that? I mean, you could have phrased it differently, but we were looking for phase 2 failures. This would be about the time in clinical trials where this would surface. If there was a problem that this technology could alleviate, this would be shown in a phase 2 trial. So we know those companies are in pain because they've already invested years and millions to this point. Last 18 months, we wanted current and so forth and so on. So we have created another dictionary with all drug companies. Contains a list, this we can reuse over and over again, all 6,000 of them. You'll see how we use that. And we also created dictionaries for failed phase 2 target organs. Oncology, we just reused it again. Another special macro we developed called Date Finder. And here's the punchline. I don't have all the answers on this just for time, but we searched clinicaltrials.gov. Great data set to find the recent breast or cancer trials that failed phase 2. And to determine the reason for these trials failing, we went to another great data set called PubMed, and they're linked. So now we can not only find out the companies, who failed what, when, why. So this is extremely important information down there. You all with me? We can come back to this in Q&A. Now, which of these companies, because now we're trying to understand the intent of that company. Is this a fly-by-night firm? Are they going to give up after this? We want to see companies that have staying power in this field of oncology, right? And so we get this. And then we come here. Now, this is the wonderful, this brings the whole presentation to a head here. Which of these same companies have demonstrated a proclivity for partnering? Where did we go to find that out? You looking ahead? Read down here. We can find their whole partnering track record from another great data set. I love the people that say stuff on the web is just junk and it's unstructured, it's got to be double junk. No, we read EDGAR for all those companies so we could actually examine their history and success at partnering. Again, partnering is using all the terms. What, you had a question, Fran?
I didn't know what EDGAR was.
Oh, EDGAR's, yeah.
It's an SEC.
That's the Security Exchange Commission of the United States, huge data set. But we've determined ways to parse it apart and read it for this type of information.
So every quarter, all the public United States capital for their 10K.
Mm-hmm.
And what you find is any event that occurred in that previous quarter that had a material impact on the company has to be listed. Often you have to find it in certain places, and then a lot of times it's in the footnotes in some of the tables. So we had to make sure we were reading the footnotes of any of the tables to find some of this activity. But you knew that later when you
We're like forensic scientists crawling all through the internet, right? Yeah, that's what it is. Now, I know you'd ask about sentiment analysis, so yes, we use sentiment analysis. Watson has a cool function already built in called sentiment. You just turn on sentiment, and it has dictionaries of 6,000 negative connotation terms, phrases, and words. 2,000 positive. It says something about the English language or our culture, doesn't it, that 6,000 negative, 2,000 positive? But it does. And that little expression right there will tell you who's a good partner. We can sift all through that. So we found out their track record, and now we're actually seeing what is said in the great blogs. These are wonderful data sets themselves out on the web about these same companies. Got it? These are short examples, but I think you get the point. We've developed a whole process for training and running Watson. It's in our book. We have other tools. I highlighted this one. Everybody, this is the workhorse we've used, but we're also, for structured data, we're in partnership with the SAS Corporation, SAS Institute, and their new platform called Visual Analytics. This will be in your, up on the site, you can have all this. This is our battery of tools where we try and fit the right IT tool to the project. I've just chosen because everybody wants to know more about Watson. I've included to give an example of that one this morning. We got a school, we got a course. We have a certificate program. All these are MBA courses today. We put a capstone executive workshop on it, and so we're actually wanting to teach companies. We teach our students. We have hundreds of grad students that can do the same thing Dick and I are showing you over the past 8 years, but now we want to go out and help organizations apply big data to creating villages. This is your favorite chart, Fran. Thank you. This is number 31. Fran sent me a note. She said, "Oh, I like this deck. I like this 31 slide." I said, "Yeah, right here. This is probably what caught your attention." Final thoughts? One more definition, again from Hargadon. I love Hargadon's words. The network is the innovation. Now think about that. Not the idea. Innovation is about connecting people and ideas. Make sense? Thank you. It is 9:54. Bye-bye. Okay. Couple questions? Do we have a chance for a couple?
Yes, we do.
Okay. Yes, we do. Yes, Dr. Ligler.
I was interested in your examples.
Yes.
And the way you made the choices of how you went down.
Yes.
So presumably some of those come from your customer. Presumably some of those come from the customer. Like, do they want to get involved with VCs? If not, then you go somewhere else for opportunities.
I'm going to help you. They all come from the customers.
Okay.
We're just facilitators in this. So we surround ourselves with the customers, the subject matter experts, the domain experts. They're the most valuable. We're just sort of the little cheerleaders right there. We have techniques for teasing out of the customers the questions and the various parts of what makes these complex questions tick, but all the real juice is supplied by the customer. Wouldn't you say that's true, Dick? Yeah, please. I just want to, we don't possess the domain knowledge. We've done 150 projects, so we're totally relying on you. But we have workshops that will pull that out, pull your knowledge out and put it in a form that we can present it to Watson. Yes, sir.
I know we kind of know the answer, but still I will ask you this. So all these big companies, when they're merging,
Yeah.
they're killing their innovation.
Right.
They know they're going to kill it, But they,
They
this
don't
is a
care.
trend. This is a trend.
Yeah.
Always, it's a business
Right.
decision
Right.
takes over
Yeah,
everything else.
yeah. Why is that?
Money. I don't know.
I'm going to put a caveat. Public companies do that.
Yeah.
Public companies,
Expectation.
could you hear his scenario? They know they're going to crush innovation. Why do they do it?
shedding innovation because then those innovative people leave. and
Yes.
form startups
Yes.
more efficiently and leaner, and then those innovations get bought back at a premium by those companies.
Right.
So it's just the free marketing work.
Nicely put.
Kelly Sexton, I love your picture of the world. It's just beautiful. It's beautiful unless you're in the R&D function of the company that's firing you. It's great.
I'm just going to,
Well, everybody doesn't have the courage to work in a startup. They don't have the skills.
our organization is based in New Jersey, where you may have read there's been a lot of exodus of the major companies. There's still, everybody still has a footprint, but not like it used to be.
Right.
And exactly what you're talking about is happening. Those that can find the resources
Right.
to get something started and have the connections back in the major companies, that going back and forth is really happening. I just want to make two comments at this discussion. One is that in my very deep research into LinkedIn, I recently searched for the term innovation director.
Yes.
And just in pharma and biotech, and there are at least 500 people with that title. Not that responsibility, but with the title. So there's a whole new.
You can tell by my grunt how impressed I
yes.
am. All right.
Yes. Well, and I've met some of these folks who are very smart people.
They're
and
very smart.
Very smart people who don't necessarily have the ears of the top brass. So, they're sort of internal champions for collaboration, but they don't necessarily have the impact, but at least it's a beginning, I'd like to say. And the other thing I wanted to mention is that this concept has been bubbling up in our organization. And Deborah Doherty, who's on our scientific advisory board, will be speaking this afternoon. And we're starting an innovation research roundtable, which will be just a small group up to about 15 people from academia and industry to start to look at what are those interesting themes that hopefully will someday develop into other programs and so on. But thank you you put a beautiful point on exactly what our organization is about, and we still have a little bit more time. So
There was one way back there. Just yell it out.
I mean, back to the previous question, what's the pressure driving these mergers?
Right.
This, presuming that they know that the innovation can be crushed, and you make the point that it's prominent public companies, is that strictly based on short-term gains from stockholder pressure?
Yes. That's my thesis, and I'm sticking it to it. Right, sticking to it. Right, yes.
So also, it's not all bad. There are some big companies.
From the school of Kelly Sexton. What's good about it?
Like J&J, the company.
Yes.
They have innovation labs, and they are going big on that. J&J.
Oh, yeah.
They have centers throughout the world now. Like Boston, California.
Yeah.
So they're doing the innovation in a different way. But they are.
Good. Probably need to. Yeah. Yes, I got the magic thing here too.
Well, I just want to thank you. I've always been a networker. I love collecting people, and I always remember people, what they do. But now I see that this can become a profession, or when your statement that the network itself is an innovation,
Right.
has just expanded my view of it. So I want to thank you for that.
You're quite welcome. That's a great way to end, because that is the main message. It's seldom the original idea or any of that. It's the network that's forged around those. Right. Here you go, sir. Do you need this, or are you wired up?
I'm going to use this.
Okay. I'm going to leave it. You want?
All right, well, it's great. I'm really delighted to be here. I'm full professor at University of North Carolina, but I spend most of my time about 2 miles from here. That's good. All right. Okay. Spend 90% of my time about 2 or 3 miles from here at the Hamner Institutes, where I direct the Hamner
University of North Carolina Institute for Drug Safety Sciences. We're now entering our 8th year. It's sort of a unique partnership between UNC and the Hamner Institutes. And I'm going to be talking today about a drug-induced liver injury, which is something I've been very interested in for a long time. And I'm reminded of when I first moved to Chapel Hill 16 years ago. My son was a teenager and was going to East Chapel Hill High School. And he came home after the first couple of days and was looking at me sort of peculiarly. And he said, "Dad, is it true that you're one of the 3 experts in the world about how a drug can hurt the liver?" And he'd been talking to the son of my chair of medicine. And I took the opportunity, of course, as being a teenager, to impress him in any way I could. And I said, "Well, you know, that's probably true, but I had to be honest." I said, "There's a catch. There are only 3 of us in the world," which was actually true. I mean, there's no medical practice, the treatment of drug-induced liver injuries. You stop the drug. So, but that all changed in the late 1990s when a drug called troglitazone, or Resolin, a drug initially made by Warner Lambert, but then as they were bought by Pfizer, turned into a fiasco. It was a wonderful drug for treating diabetes, the first drug to come along in almost 30 years, a new drug to treat type 2 diabetes. But it caused very rare but sudden liver failure and ended up being pulled off the market. There was a fellow, Melman, from LA Times, who wrote a series of syndicated articles about the corruption at the FDA to allow this thing to be approved. He got a Pulitzer Prize. It was very embarrassing. There were congressional hearings. And ever since then, liver injury or liver safety has been a very prominent thing at the FDA. They've hired a full-time person, John Senior, who all he does is look at safety databases to look for liver problems. So I'm going to talk about some of the stuff we're doing there. This just makes the point of why drugs were taken off the market over a 30-year period. Torsade de Pointe, this little thing here, major problem, but actually now there's a regulatory pathway. There are boxes you check, including a prolonged QT study that you can do that undoubtedly is going to reduce the incidence of this. But there's really been no progress in screening out in drug development the potential for a drug to suddenly cause a patient, 1 in 1,000, 1 in 5,000 or more, to turn yellow and die and need a liver transplant. There's really been no advance whatsoever. And a recent example of how sensitive the market is to liver safety issues was the experience of Sempra, our own company in Chapel Hill, antibiotic, that Reuters put out this just less than 2 weeks ago. Sempra tumbles on safety concerns of antibacterial drugs. Sempra stock plunged 37% on Friday over potential safety concerns related to the company's drug to treat a common bacterial infection that met its main goal in late stage, so it worked. The company said that while increased levels of liver enzymes were seen across treatment arms, the magnitude was higher in those who received their drug, solithromycin, relative to the comparator drug they were doing. So just a whiff. And I think the stock has since come back up, but again, the sensitivity to this issue. And what I'm going to talk to you about today is attempts to create in silico or computer-based models that can help inform companies of this potential liability. And just pointing out that in the FDA's strategic plan on regulatory science, number 3 was listed, the use to develop computational methods and in silico modeling, which I think is going to revolutionize every step in drug development in the next decade. And so what we started, we're entering our 5th year now, is a consortium. We were told to not call it a consortium because the C word is not liked in industry anymore. So we called it the initiative, the DILI-SIM initiative, and DILI is drug-induced liver injury simulation. And currently we have 13 major pharmaceutical companies and the Food and Drug Administration that are investing it. This is our village here. In addition to getting $100,000 if they'll commit to 3 years, or $120,000 a year if it's a year-by-year effort, we get their best scientists, There's a quarterly meeting by WebEx that's 2 hours, and then an all-day face-to-face meeting here in RTP of these scientists. They vote on the direction of the modeling and the projects. Food and Drug Administration actually funds one of our postdoctoral fellows, an Rees Fellowship that's here in RTP. And the goal, again, is to make a model, computer-based model, that incorporates with differential equations the various processes that underlie how a drug can hurt the liver. This is the team as it currently existed, taken at the face-to-face meeting. Particularly, Brett Howell and Scott Seiler are the two directors of this. This is our Rees Fellow being paid for by the FDA. And the great thing about these people, they really behave more like a Silicon Valley startup than an academic group. They're extremely goal-oriented, very high energy, very passionate about what they do. And we also, the other part of our community, we have an absolutely top-notch scientific advisory board for this, including David Pazetsky, who's a professor and rheumatologist at Duke, who's been a very enthusiastic, involved and supporter in this. Mark Avigan is co-director of the FDA's Critical Path Initiative at the Food and Drug Administration. The other people are all unquestionable experts. They do it for free because this helps their own research, their own things that they're interested in. Just a word on the modeling. It's what they call middle out. So we start with the problem in the liver and then model back towards cellular processes like signal transduction as much as you need to explain the phenomenon of the toxicity. And then we model forward, oh, that's shown there. Then we model forward to talk about how the drug is handled in the whole body. And then actually, as this model's created, its differential equations are over 550 statistical variables in it now. Through a genetic algorithm, we can actually vary the parameters of the variables to create a virtual patient population. And we can model a polymorphism, say in drug metabolism, et cetera. So you can make a virtual population of patients on the computer. Now, actually, the drug I mentioned that changed everything in the late 90s and put this great, intense scrutiny on liver safety and drug development is troglitazone, shown here. Again, it was first in class. Actually approved when only about 1,000 patients had received the drug for 6 months. TAC-875, a Takeda drug, which was a novel, a GP40 agonist, novel drug in class to treat diabetes. When development was abandoned late in phase 3 for liver toxicity, for rare liver toxicity that showed up, there were over 8,000 patients receiving the drug, over 4,000 patients received it for a year. Had they gotten approval, they would still need to do a 5-year cardiac outcome study. So that's a cardiac issue, but safety is really greatly expanding the size of trials, the duration of trials, and the issue isn't the cost of doing these trials, it's the time off patent. You know, if a drug's going to make $1 billion at the end of patent life, that means $3 million a day is at risk for every day you don't get approved. Anyway, this was withdrawn from the market. There was very little signal in clinical trials. Only 2% of patients developed ALT elevations, alanine aminotransferase, standard blood test for liver toxicity. There was no signal detected in routine preclinical animal studies. But it was then withdrawn from the market for rare, probably 1 in 10,000 acute liver failure, death, or liver transplant. And what was done in the model was actually to model the drug in a virtual hepatocyte. And this work was actually done by Kim Brower at University of North Carolina and a graduate student who became embedded in the team to use the model to model this. And what they did is, this is the liver cell here. And the bile is made in this bile canaliculus, which ends up going to bigger and bigger tubes and being dumped into the intestine. It's known that bile acids, the things that help in digestion, are taken up into the cell through a transport protein called NTCP, and they're then pumped into bile with another transport protein called BSEP, but some of it squirts out back into circulation through 2 transporters, MRP3, MRP4. And what they were able to show is that troglitazone is actually, when it's taken up into the liver cell, is converted largely to a metabolite called troglitazone sulfate, which potently inhibits these efflux transporters here, BSEP, and you can actually, there are companies like Solvo that you can send your drug to, and they've expressed these transporters, and you can actually measure the ability of the drug or metabolite to inhibit these transporters quantitatively. And they took that data, and then what happens when that occurs is the bile acids build up, they're toxic, causing hepatotoxicity. Now it's complicated because it turns out the parent drug also inhibits the uptake transporter. So this is a perfect situation for modeling. So what the graduate student did was take quantitative values for the ability of the drug and the metabolite to inhibit these various transporters, and then looked at the pharmacokinetics of the drug and the distribution of the drug between blood and liver through experimental studies, and actually put it into a model that already existed, that had been developed by the DILIsym team, and pressed a button, and lo and behold, predicted the actual incidence and severity of liver chemistry abnormalities observed in the clinical trials. And this was recently published late last year in a journal of Clinical Pharmacology and Therapeutics. Kung-Hee Yang was the main author, and Kim Brower. And they predicted the incidence and severity and latency of serum ALT elevations observed in clinical trials based only on the ability of the drug and the metabolite to interfere with these transporters. So no animals. This is animal rights activists love this approach. There was an accompanying commentary from the FDA where the final sentence was, "We look forward to future efforts to apply this model for prediction of hepatotoxicity that's not yet been observed clinically." In other words, we were looking backwards to do this forwards. And actually, this is from the Office of Clinical Pharmacology and Translational Sciences at Cedar. So we're very pleased about that. And actually, they're the ones now, this group, that are actually funding the postdoc in our lab. So that's one major application, is to understand why a drug causes liver toxicity and to be able to predict that before it goes into man. The other area, let me just go back here. The other area turns out to use traditional and more novel blood tests now to get more information in a clinical trial. And this I'm going to tell you the story of a drug called Intelamode, and this is a biologic single injection drug that was shown in monkeys to reduce the incidence of death after fatal radiation. And a company went to the FDA with this information and said, "We think this should be approved for the treatment of fatal irradiation," so Fukushima or terrorist action. And of course their goal is to get the US government to get several million of these syringes, and the catastrophe occurs, the helicopters come in, men in suit come out and inject anything that moves. And the FDA agreed, according to their animal rule, that the monkey data was sufficient for efficacy, but they still had to show safety in healthy volunteers. So they gave the injection, started giving the injection to a planned safety study of 100 healthy volunteers. But what they saw was that some people, this is time after the single injection here, the blood levels of this protein, which is the common, most sensitive, it's a biomarker that's been used for half a century, on liver injury. It's contained in liver cells. And there were some patients, or some healthy volunteers, that had very high ALT elevations, including one individual where the serum ALT went over 1,000. To put that in perspective, by convention, serum ALT greater than 3 times the reference range or upper limits of normal is considered clinically significant, and it is recommended in clinical trials, phase 3 clinical trials, that you stop treatment when it goes 8 times. So these were healthy volunteers. The study was immediately put on hold. The FDA made the comment, the medical examiner said, well, to tell you the truth, if I get fatally irradiated, I take an injection of this, but there's no way to take this forward because it's clearly unsafe to get safety data in human beings. And there will be other treatments that'll come down the line, and we have to be able in the future to weigh the risk and benefit of these things. So what the team did, noting that this is a very rapid rise and a very rapid fall, used the model to actually calculate what percent of liver cells had actually burst open and released their content to cause this pattern, the pharmacokinetic pattern of this blood test. And so they actually took the highest guy, they took the 95th percentile, and then the median, and they fit that in the model, and then they used the model to say how many cells must have burst open and released their contents. And what they showed, although this was an alarming level, even the peak volunteer, it was less than 3.5% of their total hepatocytes that had caused that. They then said, well, the literature says the amount of this protein per liver cell can vary, and they had literature-based variation on that. The half-life, once it's released into circulation, is as little as 31 hours, as much as 52 hours. So they had to put that variation in there. And then they also looked at the ability of the liver to recover, et cetera, but put this in this virtual patient population. And then came up with a range of percent hepatocyte loss for any peak serum ALT, again, assuming the same pharmacokinetic profile. And they showed even in this virtual population, the person with the highest ALT was still less than 5% of their total hepatocytes. Now, I can give 40% of my liver to my mother. It's called liver transplantation. I forget orthologous liver transplantation, that's what it is. And I'm a healthy volunteer, that's considered ethical to do that. Who is the FDA to say somebody can't give 5% of their liver, and the liver's like a tadpole's tail, it grows back, to save humanity or save the city of New York? So this was presented to the FDA. And the FDA didn't say much, they listened to this. However, they are allowing the company to go forward, but they said, what would be the peak serum ALT that you would definitely have to stop? And they could have said, well, how about 2,000? But instead they said 10% of the liver, and used this model to extrapolate what that peak serum ALT would be, assuming the same profile. Now, this does assume necrosis. It assumes the liver cells are bursting open, releasing their content. There are other types of cell death. And it turns out there are now blood tests that can inform how much of the cell death is necrosis versus apoptosis. So here's a liver cell releasing into blood this ALT, the thing I was talking about. Also, there's a whole revolution now in microRNAs as biomarkers. Turns out the liver is the only organ that has one of them called miR-122, so it's very specific. But then in necrosis, you release something called full-length cytokeratin 18. In contrast, in apoptosis, you release a caspase-cleaved fragment of cytokeratin 18, and you release less ALT and less miR-122. And you can actually estimate the percent necrosis versus apoptosis with the ratio of the full length to the cytokeratin 18 fragment. It's called the apoptotic index. Now, these are not the liver specific, but you can actually translate this or key it to the amount of liver injury by actually looking at miR-122, and this is all in the model. So it's now possible to estimate from serial blood sampling the percent of hepatocyte death and chance of encountering irreversible injury. And this has been part of 2 regulatory submissions, one at the FDA and one at the EMA in Europe. And I can say it didn't, one of the modeling efforts did not appear to convince the FDA, at least yet, that there was less risk than they thought. However, it did convince venture capitalists to invest in going forward with the drug. So anyway, I think within 3 years, we're well positioned that when a potential liver safety signal is identified in a clinical trial, I think regulators are going to routinely ask, what did the DILIsym model show, this model that's being created? And there has been increasing interest in our partners in the initiative initially, but now outside the partners, to do proprietary projects. The initiative is a public-private partnership. Everything gets published. Everything's in the public domain. So a company was founded based at the Hamner called DILIsym Services, Inc. in July 2015. They already have $1.5 million in contracts, and it's going very well. And I think it's an interesting business model because the actual intellectual property is being created by the major pharmaceutical companies. They're directing the development of it. So there's really no need for venture capital here, at least there hasn't been, because we have an engine. 8 of our partners have agreed to fund through 2018. And I thought the fact that a company was being created might be a negative. The idea we're providing venture capital actually was a positive to the companies because they want this widely available, widely accessible so that it'll be accepted. There is no regulatory path for acceptance of a model like this. It's simply if the FDA starts using it, it's in. So this is, I talked about the in silico modeling. Actually, we've developed a bunch of other programs, including with a guy named Ed Lecluse, liver cell culture models. And our partners in the initiative have asked us now to develop a proposal and a budget to do something similar in the various academic and commercial models of the liver, to see which ones are fit for purpose for identifying the key pathways and other things and consider metabolites to feed into the model that we've created. So just to say thanks, these are various people. I won't walk through it, and I'm done. Thank you.